Hey, it's Jeff Hansen again. I hope you remember that the very first day of class, the first thing I said is, hey, hit the ground running. This has been a very fast course. There's only a couple weeks left. A couple weeks is still a lot of time to learn a lot of biochemistry and to do well on the final exam, which is worth about as much as the first two tests combined. I know a lot of you have picked up a lot. I can tell from the conversations on the phone and with Zoom meetings. There's still a lot to be learned. My hope is that you take your time with this video, work your way through it. In part, what I've done is I've reviewed what I already covered when I introduced carbohydrates when I was just talking about the polymers of life. But in addition to covering some of that again to remind you of where we're coming from, I've extended it and gone into more detail. So yes, I think every one of you knows exactly what an anomeric carbon is, how to find it, the difference between alpha and beta glucose is. But now we're going to use that information to build up and understand polymeric forms of glucose, as well as to understand other carbohydrates. I'm hoping you're working on this over the weekend, getting ready for the final exam. And if you have questions, don't hesitate to call. Let's see how much biochemistry we can learn in a 2-month summer course. So let's begin. What is a carbohydrate? What differentiates between a carbohydrate, an amino acid, and a hydrocarbon? These are profoundly important biological molecules. For example, can you classify each of these? I don't think you'll have a problem. But it's not just classifying them, even though you're aware of them. I'd like you to interpret the name. An amino acid has an amine and an acid. A carbohydrate has carbons in it, hydrogens in it, and oxygens. 8 pretty much implies oxidized or oxygen, and usually 8 means that it's a deprotonated carboxylic acid, although it doesn't have to be, as in carbohydrate. And then hydrocarbon, hydrogen and carbon, because fatty acids are almost completely hydrogen and carbon atoms with only a single carbon that's been triply oxidized, a lot more hydrogen carbon and carbon to carbon bonds by weight in a hydrocarbon than in a carbohydrate. And let me throw in the hydrogen atoms just to remind everyone that every time you see a bend in one of these molecules or an end, that means there should be hydrogen atoms. So here where I have the terminus of the hydrocarbon, there should be 3 hydrogen atoms, and every bend in the molecule, there should be 2 hydrogen atoms added. So again, carbohydrate. They're made from carbon, hydrogen, and oxygen. Interestingly, hydrogen is something that can be generated from water. You don't have to memorize that hydrogen is generated from water, but the language of chemistry and biochemistry is so intricately tied together and so systematic that the better you understand it, the easier it'll be to use. Most of you have probably seen this figure in a biochemistry textbook where you have every triose, tetrose, pentose, and hexose that's possible. I have to say that please don't bother to learn this tree, although absolutely you should know that tri is 3, tetra 4, penta 5, and hexa is 6. The Fisher diagrams give really clear information on stereochemistry, but it would be foolish to memorize the orientation of each hydrogen and hydroxyl in such a random figure. So what do I want you to know from this? Well, there's a handful of really important sugars that you do need to know about, such as glyceraldehyde, because glycerol is a 3-carbon alcohol, glyceraldehyde, a 3-carbon sugar. You're going to hear a lot about glycer. Glucose, galactose, and fructose, these 3 hexoses at the center of life, at the center of our diets, and at the center of our metabolism. You really want to get to know them well. And ribose is pretty important too if you want to have ribonucleic acids or deoxyribonucleic acids in your genome. In the figures I use, I won't be drawing all the hydrogen atoms in. I'll be simplifying glyceraldehyde. I'll be simplifying dihydroxyacetone. And along those lines, can you apply simple organic nomenclature to assign each of these structures to each of these words? Let's practice a little of the nomenclature we were looking at in the previous videos. We know that glycer means 3 carbon atoms. Can you assign the rest to these molecules? Please mark A, B, C, and D. Glycerol is the least interesting. It's only going to be hydroxyl groups, no double bonds in it at all, glycerol. Glycer8, well, I'm looking at a carboxylic acid, and there's only one of those with a carboxylic acid. You'll notice that this is actually glyceric acid, but people usually don't differentiate whether the proton is on or off because it's going to be in equilibrium anyways. Glyceraldehyde. Well, only one aldehyde group here. This carbon atom would have only one hydrogen coming off it, and that leaves dihydroxyacetone. So acetone is the simplest ketone, a carbonyl with a carbon on each side. You hydroxylate each of those, dihydroxyacetone. In order to understand sugars, we have to be familiar with these different functional groups. You should be able to number using Greek letters and Arabic numerals, and you should know what a nucleophilic attack step is. These are things we've already covered. Now, I want to use that organic nomenclature to help you understand how to think about carbohydrates. You know nothing about carbohydrates unless you know cold what an anomeric carbon atom is and how to find that in a structural representation of a carbohydrate. There's a lot of other nomenclature that mix some of the previous organic you learned with a bit of nomenclature that's more unique to carbohydrates. For example, the ending os simply means sugar. So we have gluc sugar, fruct sugar, suc sugar, lac sugar, aldehyde containing sugar, ketone containing sugar. You're going to want to have a working knowledge of all of these names. In fact, in order to help you understand how to think about these really important carbohydrates central to diet, metabolism, health, disease, I'm going to go into greater detail than you need, but the key thing is, can you get the big, most important algorithms and apply them in some detail? You will find parts of this to be complicated, and when it gets complicated, I hope you'll contact me. I think that if you talk to me, you will find that you will save yourself a lot of time and a bit of a headache. So you run into trouble, maybe you should contact me before you try to move on in the video. Regardless, I think you'll enjoy a lot of it. But I want to begin with a question. Both potatoes and table sugar are made pretty much out of sugar. In the case of a potato, it's a polymeric form of sugar. In the case of table sugar, it's a dimeric form of sugar, a disaccharide. Whether you eat a potato or table sugar, they are very rapidly hydrolyzed down to monomers, which are very rapidly absorbed by your digestive system. So, nutritionally, are they equivalent? Please pause and explain. And I'm going to say, my answer is absolutely not. They are extremely different because the potato is made out of starch, which is pure glucose. In the previous class, I'm now near the end of year 1, and a number of medical students still don't recognize that starches, which are half of our dietary energy, whether it's in the form of wheat or corn or rice or potatoes, is pure glucose, but in a polymeric form. In contrast, table sugar is a dimer, but it's not made of pure glucose like potatoes. Rather, it's a dimer between glucose and fructose. Therefore, if you eat a baked potato, that starch will rapidly be digested, and in your intestine, you will have a massive amount of only glucose monomers. However, if you take in the disaccharide of table sugar, it also is digested very rapidly, and in your intestine you will have a very different mixture Half of it will be the same glucose as you get with a potato, but importantly, the other half of this will be fructose, which is actually harmful. In the case of the potato, as it gets completely digested down to solely glucose, your intestine absorbs it, it gets into your blood. Insulin and glucagon regulate the uptake of glucose into cells, whereas insulin and glucagon don't regulate fructose. Inside of the cell, glucose metabolism is carefully regulated between the different needs of the cells. However, fructose is not regulated among the different needs of the cell, which can contribute to metabolic diseases such as non-alcoholic fatty liver disease. In fact, Americans take in so much sucrose, half of which is fructose, that I believe at this point we should define fructose as a toxin. I'm going to be talking about regulating glucose and its central role in brain metabolism, red blood cell metabolism, muscle metabolism, in basically every course this year and next year. I also will be talking about fructose in a lot of these courses. For that matter, the physiologists and clinicians and probably the immunologist and microbiologist will also be talking about the many roles that fructose plays in diseases, such as diabetes. How did we get here? For the past 10,000 years, our diets have been very stable. Most of our calories have been taken in the form of carbohydrates, and specifically in the form of starch, potatoes, rice, wheat, corn. It was only after they started purifying sucrose from sugar cane and sugar beets that the amount of sucrose and of fructose in our diets began to grow exponentially, especially over the past few decades. Now, to be clear, for the past 10,000 years, half our dietary calories have come from carbohydrates, about a third from fat, about a sixth from protein, and that's the way it remains till today. However, previously, those carbohydrates were almost completely in the form of starch, pure polymeric glucose, and maybe 1% of the carbohydrates was due to sucrose, which is found in various fruits and vegetables. Who knows, a treat of a little bit of honey here and there. It was only when they started to purify sucrose from sugar cane and sugar beets that we began to increase our intake. Now, in the typical American diet, 50% is still carbohydrate, but the fraction of that that's in the form of sucrose has grown to about 20% of dietary calories, such that 10% of dietary calories are in the form of fructose, the unregulated renegade carbohydrate has become a macronutrient. So over the past few decades, there's been a dramatic change in the nature of our carbohydrates. Specifically, our intake of fructose has been growing exponentially. Interestingly, in the same time frame, the epidemic of obesity has been growing exponentially. And the epidemic of diabetes has been growing exponentially. It seems remarkable to me that since 1960, the incidence of diabetes in America has grown eightfold. It went from about 1% of Americans up to over 8% of Americans currently. Many studies showed a correlation between the increased incidence of metabolic diseases such as diabetes and the increased consumption of sugar, shown in purple. And by sugar, I mean specifically sucrose, fructose, and glucose, those small sweet molecules. I'm not talking about the giant polymeric form of glucose, starches. Those studies were trying to show that, hey, this increased intake in sugar is causing all these metabolic problems. And I want to be real clear here. The take-home lesson is, yes, our dietary changes with respect to carbohydrates have undoubtedly been harmful and are harming your patients right now. However, I have to point out the story is more complicated, that there are other factors involved, other components of our diet, of our exercise, of our lifestyle. And for that matter, Correlation does not prove causation. One exponential growth rate looks like another. In the same time we saw these exponential curves, the stock market was growing exponentially. Human population was growing exponentially. The cost of a home in Gilbert was growing exponentially. And for that matter, the correlation began to break down in around the year 2000. So at that point, Americans evidently started listening to their physicians and to the scientists and decreased their consumption of sugars by about 1% a year, decreasing a total of about 14% of their total intake. And yet we see that exponential growth rate for diabetes has not yet been seen to decrease. So again, the story is more complicated, but undoubtedly our diet and the sugar component of our diet is important and is harming us. Because of the central role of carbohydrates in human metabolism and human diseases, I will talk more about carbohydrate metabolism over the next 2 years than any other topic. In addition, the physicians are going to talk extensively about diabetes and about dozens of different metabolic diseases caused by loss of function mutation of the many different enzymes which are involved in sugar metabolism. The easiest way to learn and memorize all those different enzyme names and all of the carbohydrate metabolic pathways involved in those diseases is to begin by understanding the simple nomenclature of carbohydrates based on their structures. Your choices are memorize dozens of enzyme names and dozens of pathways as gibberish, or learn first how to understand the simplest sugars, the monosaccharides glucose and fructose, and the disaccharide sucrose. And to be clear, I'm not asking you to memorize these structures. I am asking you to either memorize or understand some general concepts about their structures so that then you can use them to build into all these complicated pathways and diseases. And you'll be able to understand the nomenclature that the physicians and I will be using over the next 2 years. For example, first of all, what would you name the enzyme that hydrolyzes the disaccharide sucrose into the two monosaccharides, glucose and fructose? That much nomenclature it's worth memorizing, and you're going to build from this type of nomenclature to the higher levels of complexity in terms of pathways and diseases. What do you call the enzyme? You could have said many things and you wouldn't be wrong. Technically, the name is sucrase. If you said, let's see, it acts on sucrose, so the first name is sucrose substrate. What does it do? It hydrolyzes it. How about sucrose hydrolase? Perfectly good name. And you don't have to worry. If that's what you came up with, I'm pretty happy. Because sucrase hydrolase could have been the name of it. It's not. And you can only put on a board exam real enzyme names. Therefore, sucrose hydrolase will not be a distractor on exam. And therefore, I think you'll be able to interpret sucrase. A is the enzyme that acts upon sucrose. So interpretation is where it's at, not memorization. What other structural component of glucose and fructose do we need to understand? At the heart of these sugars is the fact that they are in equilibrium. Glucose is in equilibrium between a cyclic and linear form, and fructose exists in equilibrium between a cyclic and linear form. That you have to know cold. Also, you need to know which is a ketose and which is an aldose. Please pause and tell me. Again, basic organic nomenclature. Ketose, os always means sugar. Ket is from ketone. Aldose, os is from sugar. Al is from aldehyde. Which is an aldehyde, which is a ketone? Aldehydes and ketones are based upon a carbonyl group. A carbonyl being a carbon-oxygen double bond. What distinguishes them is that in the aldehyde, the carbonyl carbon is at the end of a carbon chain such that adjacent to it will be a small hydrogen atom. In contrast, in a ketone, the carbonyl is not at the end of the carbon chain. Rather, there's a carbon atom adjacent to the carbonyl on both sides. How will that affect the reactivity of aldehydes versus ketones? Please answer. Do not memorize one is more reactive than the other. Think about the logic behind it. Well, when we're talking about reactions, what is the mechanism we're always thinking of? A nucleophilic attack. So a nucleophile will need to get access to the carbonyl carbon. How will it do it? In the case of an aldehyde, there is a big carbon chain that will get in the way somewhat, but on one side of the aldehyde is a lone little bitty hydrogen atom, so it's much easier for a nucleophile to get access to the carbonyl in an aldehyde than in a ketone. In a ketone, the carbonyl carbon is surrounded on both sides by large carbon atoms. On one side, an entire chain. On the other side, at least one carbon with oxygen and hydrogen atoms. Therefore, it is harder for the nucleophile to get into that narrow opening in order to attack the carbonyl carbon. At this point, I'm sure some of you are saying, I don't quite get the chemistry, so I'll just memorize that an aldehyde is more reactive than a ketone. That would not be a good idea. Certainly you can use brute force memorization to learn almost anything short term. I'm quite sure most of you will suffer some type of cognitive overload at ATSU University because some things you just do have to memorize. You can reduce that burden greatly by not using brute force memorization when you have the alternative of understanding the concept, especially when it is so broadly applicable. Why is this broadly applicable? Why do you think the FDA says you shouldn't drink formaldehyde? It is that extreme reactivity of the aldehyde group that's so toxic throughout your body. And for that matter, how about the aldehyde group on glucose? It is that reactive aldehyde group on glucose which makes the hyperglycemia so harmful in chronic diabetes. That aldehyde group reacts with many tissues. In the eyes, it leads to diabetic retinopathy and often blindness after long-term diabetes. In peripheral nerves, it leads to diabetic neuropathy, which is the number one cause of non-traumatic limb amputation. And in the bones, it glycates collagen, weakens the bone, and leads to a higher incidence of bone fracture in people with diabetes. So the chemistry of that aldehyde group is clinically very important. And to understand that chemistry, as well as the nomenclature of the enzymes and the products and substrates of the metabolism of glucose, it helps to know the numbering of the carbons. WU applies the rules of nomenclature to number each of the carbon atoms in linear glucose and in linear fructose. Please pause and do that. Let's look at glucose first. Unlike a fatty acid that only has oxygen atoms attached to one carbon, here we have an oxygen linked to every carbon atom. Therefore, we need to find something unique about one of the atoms in order to find a landmark. What is that? I think most of you know. If you don't immediately know, take a look and see if you can figure it out. And the answer is that, one of the oxygen atoms is different. It's in a double bond. And two bonds to the heaviest atom is more important than one bond to the heaviest atom. So in sugars, it is that carbon atom that's bonded twice to oxygen that's important, and it is extremely important. This is probably the most important learning objective in the entire presentation. This is called the anomeric carbon. If you can't find the anomeric carbon in a sugar, you're wasting your time. And the principle is simple. You don't really have to memorize anything because it is clearly distinct from the other oxygen atoms. Okay, so now we have the anomeric carbon. Number all of the carbon atoms in glucose. Well, using the landmark anomeric carbon, it's easy. 1, 2, 3, 4, 5, 6 going to the far end. Now, how about numbering fructose? Again, with any sugar, the most important first step is to identify that anomeric carbon. Please do so. Pretty simple. Here it is, the carbon with a double bond to oxygen. Now can you number? And of course, there's a little bitty trick there, not too bad, but hopefully you got that. You don't start at the anomeric carbon, because then what direction do you do the numbering in? Rather, you start at the end closest to the anomeric carbon and numbered toward the opposite end. And so accordingly, in glucose, the anomeric carbon is carbon 1, and in fructose, the anomeric carbon is carbon 2. Will you now number the oxygen atoms in the linear form of glucose and fructose? Please pause and do so. And again, I'm trying to show you there's no memorization. The numbering is exactly what you'd expect it to be. How would you logically number the oxygen atoms? I think every one of you would come up with what the organic chemist came up with. You would number the oxygen atom based upon the carbon atom to which it is attached. And again, I want to be clear, please do not even think about trying to memorize these Fisher diagrams with a hydrogen on one side and oxygen on the other. That would be a complete waste of time. We are going to look at the stereochemistry of the individual carbons, but it'll be based on a really trivially simple algorithm based on the conformational figure, not on the Fisher diagrams. What you do need to know about the Fisher diagrams is the anomeric carbon. The anomeric carbon of glucose is carbon 1. The anomeric carbon of fructose is carbon 2, which makes glucose an aldose and very reactive, and fructose a ketose and less reactive. Which of course means that if you're looking at the stereochemistry of glucose and fructose, they're almost identical. It simply has been the movement of the double bond from carbon 1 to carbon 2 that distinguishes these. And so even though glucose and fructose are chemically very similar, the same number of carbon, oxygen, and hydrogen atoms, very similar stereochemistry. When you do move the double bond, you have the same numbering for the carbon and oxygen atoms, equally water soluble, and yet glucose and fructose are profoundly different biologically, especially with regard to human health. Whereas glucose is half of our nutritional energy, the primary fuel for your brain, the only fuel for your red blood cells, profoundly important in muscle metabolism and, for that matter, liver metabolism, metabolism throughout the body, and involved in all sorts of human diseases. Accordingly, you may not be surprised to hear that a majority of my talks over the next 2 years are going to involve glucose, and a lot of them also will involve fructose, which actually is a toxin in terms of our current diets. And I do hope that so far my detailed description of the chemistry of glucose, fructose, sucrose, and starch has already expanded upon your understanding of the health differences between a potato and table sugar. And now I want to zoom in and look at the difference between table sugars and high fructose corn syrup. In fact, you may be somewhat familiar with the controversy. On one side we have sucrose, which would include the sugar beet farmers and the sugar cane farmers. On the other side we have high fructose corn syrup, where we have a lot of the corn growers. Both sides have spent billions of dollars in advertising trying to convince Americans that their form of sugar is somehow better than the other. For example, the sucrose industry bills itself as wholesome, natural sugar, even though it's sold as a highly purified white powder. In contrast, the high fructose corn syrup has their own storyline. In fact, I think that both the corn syrup producers and the people who make what they call wholesome natural sugar, or sucrose, has become so political. They've spent massive amounts of money counter-advertising. So I hope you'll pause this video now, open the PowerPoint, and click on the link on this slide just to see the advertisement. It really, I think, emphasizes the importance of simple chemistry, sucrose versus glucose and fructose. In summary, I do agree with the sucrose industry that high fructose corn syrup is harmful. On the other hand, it turns out that sucrose is equally harmful. Both sucrose and high fructose corn syrup are made of small molecules, sugars, which very rapidly diffuse through the digestive system, get into the intestine where sucrose is hydrolyzed by sucrase into glucose and fructose, which are readily absorbed. Your body can't tell the difference. In fact, it's my opinion if there is a difference, if you have to choose between sucrose and high fructose corn syrup, I believe that sucrose is actually slightly more harmful medically than is high fructose corn syrup. And I usually find that a few students will disagree with me on this concept, and I'm delighted. There should be a diversity of understandings of how metabolism works. Now, on this particular issue, I think I'll be able to convince you that I'm right, but I always learn from the different perspectives I get from different students because diet is an extremely complicated subject that I don't think is understood by dietitians, nutritionists, nor even biochemists. I hope you will learn a lot from me relative to how biochemistry helps us understand nutrition. I also usually learn from the students, especially those of you who are interested in diets. But there are certain rules which are clear, and those are the learning objectives for the talk. Among those principles is that glucose and fructose have very different physiological effects, even though chemically they're very similar. For example, glucose entry into metabolism is highly regulated by phosphofructokinase 1. In contrast, fructose bypasses that enzyme and pretty much is unregulated. The release of insulin is stimulated by glucose, but it's not directly stimulated by fructose. Most glucose passes through the liver and is absorbed by the workhorse cells in the body, muscle, and fat cells. In contrast, fructose is almost 100% absorbed by the liver on that first pass, and liver and muscle cells don't even have receptors to take fructose in. But right now, I really want to zoom in on this profoundly important nutrient, glucose. And I've already mentioned how glucose interconverts between a linear and cyclic form. Right now, I would like you to pause the video to describe that reaction. Will you please draw arrows to represent the intramolecular reaction by which linear glucose cyclizes to form cyclic glucose? If you need a clue, it's going to involve the one mechanism I always talk about. There will be what? Yeah, a nucleophilic attack, which should tell you where the attack will occur. Please go ahead and do that. So the first thing I'm going to do is I look for that double bond. Where's that important anomeric carbon in the cyclic form? What simplifies this problem is to realize that the anomeric carbon will have two bonds to oxygen, whether it's in the linear or cyclic form. That is, the nucleophilic attack on the carbonyl carbon will cause the electrons to resonate up onto the oxygen atom, and it'll grab a proton, but that means that the linking oxygen and an exocyclic oxygen will both be linked to that carbonyl. So we look at the cyclic form to look for which of the carbon atoms is bonded to two oxygen atoms. Please do that. It's in your interest. It is absolutely in your interest to spend a few minutes here to understand this, and you'll remember this very long term. I look at this carbon here, right? And I go, okay, there's one oxygen atom here. Let's go to the left. Oh, a carbon here. Go to the right. Oh, there's a carbon here. There's a hydrogen atom going straight down from it. Not anomeric. Another clue is that when the oxygen atom nucleophilically attacks the carbonyl, that means that the linking oxygen in the ring is going to be attached to the anomeric carbon. So we know it'll be next to the ring oxygen. We can look at the carbon adjacent to the left, and we can look at the carbon adjacent to the right. The carbon atom on the left is bonded to the ring oxygen, then it's bonded to a carbon atom here, a carbon atom here, a hydrogen below it, only one bond to an oxygen atom. Go to the carbon atom on the right, go straight to an oxygen atom. The anomeric carbon is linked to the ring oxygen and to an exocyclic oxygen atom, so therefore you've identified the carbonyl carbon. So to summarize, in thinking about going from linear to cyclic forms of sugars, first of all, in the linear form you look for a double bond. In the cyclic form you look for two single bonds to oxygen, so that either way the carbonyl carbon has two bonds to oxygen. In addition, by default, the anomeric carbon should be on the right side of a conformational figure. So that's also a clue, but always look for it being next to the ring oxygen and having two bonds to oxygen atoms. And now that we can see how the ring formed, will you number the carbon atoms first in linear glucose and then also in cyclic glucose? Please go ahead and do so. But I need to move on. With linear glucose, that's very easy. We simply look for the carbonyl 1 through 6 to the opposite end, right? Now with cyclic, it's a little more difficult. However, we know that carbon 1 is the anomeric carbon, and if we're looking for a continuous carbon chain, we can't go around the circle on the top here because there's an oxygen atom. The only way you can number is to the left on the bottom here, 2, 3, 4, 5, 6. And again, since the nucleophilic attack on the aldehyde group happened from carbon 5, that means that carbon 6 remains exocyclic. It's not in the ring, but it stands out, which makes that hydroxyl a little different from the other ones. An even more important learning objective is that cyclization of linear glucose leads to two different conformations, alpha glucose and beta glucose. This occurs because the nucleophilic attack of oxygen 5 on the aldehyde group can either occur from above the ring or from below the ring, which will push the hydroxyl group either up above the ring of the glucose or down below the ring of the glucose. And the difference in stereochemistry between alpha and beta glucose plays a really important role that differentiates between half of our dietary energy, digestible starches, and one of our most important dietary fibers, which is indigestible, called cellulose. Accordingly, understanding human nutrition makes more sense if you understand the difference between alpha and beta glucose. For that matter, a number of enzymes which are implicated in metabolic diseases because of the way they interact with carbohydrates are named based upon the alpha and beta nomenclature. Since alpha and beta glucose lead to very different biology, this again is something I want you to memorize. And this is one case where I don't resort to pure logic or pure systematic nomenclature. Rather, I resort to a trick. I'm embarrassed, okay? But we have to do it. The simplest algorithm I know about is to say that alpha is like a fish, and it swims below the plane of the glucose, and beta is like a bird that flies above the plane. And the reason I mention this is I myself have never found a logical way to memorize this, and I think as a physician, a very simple algorithm that will help you understand the names of a number of different enzymes and how starch works versus how cellulose, the fiber in our diet, works. So again, this is a learning objective. You will be quizzed on it. If you can come up with an easier way to remember it, please go with it. To help you become more familiar with this very important alpha versus beta nomenclature, I would like you to look at alpha and beta glucose and tell me which of these you think will be most prevalent in human blood. Please pause the video and answer the question now. However, I'm moving on. The question about which is more prevalent really means which has a higher and lower energy. The lower energy something is, the more prevalent it'll be. The higher the energy of something is, the less of it will form. So what's the difference in energy between these two molecules? The only difference structurally between these two molecules is the stereochemistry at the anomeric carbon. One hydroxyl points up, the other points down. Now, low energy generally means being more spread out in space. When you have large atoms like oxygen and carbon, the more trans bonds there are, that's the more often the backbone reverses, up, down, up, down, up, down, the lower the energy. Anytime there's a cis bond where you go up, down, down, there's a cis bond, those large atoms are crowded together and it is going to occur less often. So which of these is all trans? Is one of them cis? Please take a look and see. Let's start with beta-D glucose. First, we'll look at the atoms on the top of the cycle, starting with the hydroxyl. Up, down, up, down, up. Every bond is trans. Let's look at the bottom of the cycle. Up, down, up, down, up. Every hydroxyl group in beta-D glucose is equatorial. Now you can look and say, hey, what about this oxygen on carbon 6? Well, you realize that's not directly linked to the ring. Rather, there's a linking carbon group, so this hydroxyl is free to rotate. The linking carbon, carbon 6, is equatorial, as are all the hydroxyls. In contrast with alpha glucose, let's do the same trace starting at the top of the ring. Up, down, down. Let's look at the bottom of the ring. Up, down, down. The hydroxyl coming off the anomeric carbon in alpha glucose is cis. It tends to bump into this ring more than the equatorial oxygen on the anomeric carbon of beta glucose. And in fact, let me emphasize that by just pointing in red a tracing showing everything is trans going across the top of beta glucose, whereas there's the one cis bond in alpha glucose. We can also look at the bottom trail. It's always the same. A cis bond is going to be higher in energy, and as a result, in your blood right now, about 2/3 of the glucose molecules is in a beta glucose, a cyclic form. About 1/3 is in alpha glucose cyclic form, and less than 1% is linear. This is kind of fortuitous. This aldehyde group is very reactive, and as you age, that aldehyde is going to interact more and more with more tissue in your body, or if you have diabetes and have extra linear glucose, it'll interfere with things. It's nice that over 99% of your glucose, that reactive aldehyde has interacted intramolecularly and is now inert, held into the cycle, which can float around and bring nutrition throughout your blood at very little expense. This nomenclature that I've been using, where I refer to hydroxyls as equatorial or axial, is really important because you do not need to memorize a single Fisher diagram if you have a conformational figure. You can absolutely identify glucose based on stereochemistry in a conformational figure based on applying a single algorithm. You want all of the hydroxyl groups to be equatorial. You want all of the heavy atoms linked through trans bonds, with one exception. And what's that? Please answer. The one hydroxyl which does not have to be equatorial is the hydroxyl linked to the anomeric carbon, because that can point either up or down. It's anomeric. It doesn't have a specific stereochemistry. In a conformational figure, glucose makes perfect sense. Glucose has the lowest conformational energy of any hexose, because every single hydroxyl in it is equatorial, pointing away from the mass of the ring, and only these small hydrogen atoms are pointing axially, straight up and down. They can bump into the ring, but they're so small they don't contribute much to energy. And in fact, this equatorial versus axial nomenclature is so important. Let me go ahead and throw the earth up here. If you have any question about it, please do contact me. Applying your current understanding of glucose to these figures, will you identify which of these sugars is glucose? Please go ahead. At this point, I believe you should find this question trivial. You simply apply the one algorithm that glucose is going to have the lowest possible conformational energy. All of its large atoms should be in trans bonds. All of the hydroxyls should be pointed equatorially away from the central ring. So let's look at the first example. We can see, let's see, is equatorial, which is this, which is this. These are all equatorial. Wait a minute. What about this hydroxyl? Do you see how it's pointing vertically straight up? This is an axial hydroxyl. It's not the anomeric carbon at the opposite side, at the right hand of the figure where it should be. So in fact, absolutely not glucose. How about this one? Equatorial, equatorial, equatorial. Never worry about carbon 6. Oh, hey, this is an axial hydroxyl. Is this glucose or not? Ed, I hope nobody's tricked by that. This is the anomeric carbon. It's linked to two different oxygen atoms. It is axial, but that's allowed. Which form is this, alpha or beta? Let's see. The hydroxyl is below the plane, swimming like a fish. It's an alpha. How about this one? Well, right away you probably noticed we have an axial hydroxyl on carbon 2. This is not glucose. How about this one? Equatorial, equatorial, equatorial. Everything's equatorial. This is the lowest possible conformational energy. Not only is it glucose, which form of glucose is it? The beta form. And to confirm that, here we have the hydroxyl above the plane of the glucose, flying like a bird for beta. Now, I do want to go one step further. This is a slightly lower priority learning objective, but can you identify the first sugar in this figure? Please take a stab at that. Well, we said it wasn't glucose, and we see that what carbon number has the axial hydroxyl group? Well, in the case of glucose, the anomeric carbon is carbon 1, 2, 3, 4. So carbon 4, you'll notice, is on the complete opposite side of the ring as the anomeric carbon. And it turns out that the monosaccharide galactose is identical to glucose except at carbon 4, where its hydroxyl has the opposite configuration, which would put it in this axial position. And we will talk a lot about the dimer lactose, lact from lactation or milk. Lactose is the sugar in milk. And when you break the dimer apart, you get glucose and galactose from lactose. And again, I think it's a learning objective for now. It'll be more important when you get here to AT still. And I just want to point out that the central sugars I've talked about, galactose, glucose, fructose, and glycerol, each of these enters central metabolism through a different entry point. Now, you don't have to worry about this figure now, but when you get to AT Still University, we will be looking at it one piece at a time. So I've spent a lot of time talking about monosaccharides and disaccharides, glucose, fructose, sucrose, high fructose corn syrup, galactose, glycerol. And now I want to go and describe in more detail polymeric forms of carbohydrates. In order to do that, I would like you to begin by trying to write a short definition for each of these terms related to carbohydrate nomenclature. You probably have a good feeling for the fact that carbohydrate is the encompassing category that includes sugar, starches, cellulose, which are compounds containing carbon, hydrogen, and oxygen in a ratio of approximately 1 to 2 to 1. Hence the formula CnH2ON. Sugars are a subcategory of carbohydrate referring to small molecules, monosaccharides and disaccharides that are small enough to bind to taste receptors and often taste sweet. Starches, in contrast to sugars, are huge molecules, way too big to bind to taste receptors so they don't taste sweet. They are polymers of specifically glucose. And we'll talk shortly how those glucose molecules are linked in 1,4 alpha-glycosidic bonds, and there are occasional branch points through 1,6 alpha-glycosidic bonds. And again, this is half of all dietary energy. Cellulose, like starch, is a huge polymer of glucose. Starch or cellulose can have 10,000 glucose residues in it. Unlike starch, however, cellulose is linked through 1,4 beta-glycosidic links, and it is the difference between alpha and beta that gives them that very different characteristic that cellulose is indigestible. And we're going to describe why shortly. But accordingly, cellulose is a very common form of dietary fiber. Let's take a look at these polymers. I mentioned that the glucose residues in starches are alpha glucose, and the glucose residues in cellulose are beta glucose. Let's start with the lower figure here. Can you determine whether this is alpha or beta? And can you look at each glycosidic link and tell me which carbon atoms are in that bond? Please go ahead and do that. But I'm going to move on. Now, if you had trouble, we'll zoom in on one of the links. Go ahead and characterize that. Identify the number of each carbon in the glycosidic link and determine whether each glucose residue is in the alpha or beta configuration. So, number one, let's look at the bond. The oxygen atom is here, so the two carbon atoms are bonded adjacent on each side. Can you tell which of these carbons is going to be anomeric? Please do so. Yeah, well, the anomeric carbon has a bond to two different oxygen atoms. And of course, it helps that one of those is always going to be the ring oxygen. So that kind of helps identify it. Also, it follows the rule that the anomeric carbon should be on the right in a figure if that's easy to do. So yeah, the anomeric carbon is here. So what number is the carbon on this residue? Well, the anomeric carbon in glucose is carbon 1, so this is 1. Can you tell me whether the bond is alpha or beta? Well, the oxygen is below the plane of the sugar. That makes it an alpha like a fish. What's the other carbon atom? Can you number it? We'll have to look at this residue to do that. Find the anomeric carbon. 1, 2, 3, 4. So this is a 1,4 alpha glycosidic link. Now can you similarly identify this link? Please do that. But I'm going to move on. So again, we have the oxygen in the middle, a carbon atom on each side. Which of the carbon atoms in the glycosidic link is the anomeric one? Which of the carbon atoms is linked to two oxygen atoms? Yes, it's also on the right-hand side of the monomer, as it is in standard notation. And it's linked to the ring oxygen, so anomeric is here. In glucose, that's carbon number 1. On the other side, we look at the other monomer, the anomeric carbon. Oh, now notice there's an oxygen here, so we'll have to go on the top of it this time. 1, 2, 3, 4. So this is a 1,4 beta link. So the 1,4 beta link makes cellulose. The 1,4 alpha link makes starch. And in fact, it is the nature of these bonds that allow cellulose to form extremely straight structures because every bond can be trans. Up, down, up, down, up, down, up, down, up, down, up, down. You keep reversing the backbone, you can do a very straight line. However, in starch, since every glucose monomer is in an alpha configuration, that means that there is one cis bond for every single glucose residue, which means that this is kinked at every glycosidic bond. It cannot form a straight chain, and generally it forms a curve, as shown here. Now, I'm going to go a step beyond here. What I've described so far is the most important, but I do want to go up one more level in complexity to be sure you understand this alpha, beta, fish, bird nomenclature, because you'll notice that the definition that in beta glucose the oxygen is above the plane of the ring, and in alpha glucose the oxygen is below the level of the ring, then that definition applies specifically to the standard orientation of a conformational figure of glucose, where the anomeric carbon is to the right, where the ring oxygen is above and going back into the plane. What happens then if we take the glucose monomer and flip it over such that each glucose residue in cellulose flips 180 degrees relative to the previous? And that is the actual structure of cellulose because these residues will rotate into the lowest energy conformation. In this case, as we would expect, you see that the standard representation of glucose puts the oxygen above the plane of the ring. And by definition, if you rotate that ring 180 degrees, now the oxygen will be below the level of the ring. But you'll notice that in both cases, the oxygen in beta are in the equatorial position. And of course, in alpha glucose, the oxygen not only will be below the ring in the standard orientation, but in any orientation, it will be axial. So maybe we could say that alpha glucose looks like a fish and alpha is also axial. So that's a slightly added level of complexity to the alpha-beta nomenclature. But why is it then that a straight polymer of glucose can't be digested, whereas a curved one can? These straight polymers of glucose can pack tightly together because they're so straight. It's real easy to pack things together if they're straight. If they're bent or if they're curved, you cannot uniformly pack them together this way, and therefore cellulose packs extremely tightly and extremely densely, and it's one of the most important structural components in nature. In fact, glucose is the most prevalent organic molecule on the planet in a polymer as cellulose, which means specifically that the cellulose form of glucose is the most prevalent molecule on the planet. In what type of role might these really straight, tightly packed fibers form? How about trees? Most of the vegetation you see around you is supported by a lot of cellulose fibers. In contrast to cellulose, starch, with its 1,4-alpha glycosidic links, has a kink for each glucose residue. It cannot form a straight line. Instead, it tends to loosely form into these helical structures, which are very floppy but can pack together, and they are therefore not useful structurally whatsoever. However, because they're so floppy and so highly hydrated with water, they're very accessible to enzymes, and this makes starches, with its 1,4-alpha links, extremely useful as a fuel. Enzymes can rapidly act upon it and build it up as a fuel storage, and enzymes can readily access it and tear it apart to mobilize it when energy is needed. And what I'm showing here is the simplest form of starch called amylose. Amylose, the simplest starch, is formed from linking glucose monomers through 1,4-alpha links to make one simple linear molecule. In contrast, the more complicated amylopectin is formed by taking these amylose helices and linking them together to make branch points through alpha-1,6 glycosidic links. In fact, typically there are multiple branch points in amylopectin such that there can be 10,000 glucose residues in one amylopectin starch molecule. Accordingly, amylose and amylopectin are the primary storage forms for glucose in plants. Animals, in contrast, store glycogen, which is also made out of alpha-1,4 links with alpha-1,6 branch points. So what's the difference between glycogen and amylopectin? It shows in the figure here. Take a stab at it. This is not real important. Glycogen, it turns out, has a lot more branch points than amylopectin, so that each branch tends to be shorter in glycogen than in amylopectin. The key is that amylopectin and glycogen are very similar structurally and play very similar roles in plants and in animals. That's as far as I want to go. A couple questions here if you want to review some of what we've looked at. I hope you've enjoyed this. I look forward to meeting everyone. Thank you.